أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم وله الجوار المنشآت في البحر كالأعلام فبأي آلاء ربكما تكذبان كل من عليها فان ويبقى وجه ربك ذو الجلال والإكرام فبأي آلاء ربكما تكذبان صدق الله العلي العظيم. Lecture number 23 for the first year class, and informations that enzymes are friends. They work like cricket team. They work alone and they work in the form of team as well. Just like football players, they will make plan. They catalyze chemical reactions by coordinating with each other, and each enzyme will do its own function. Some like one player is a goalkeeper, one player in forward position in cricket, one player is a bowler, one is a fielder, one is a batsman. They will allocate the duties like enzymes. They showed us how to win the match. If you want to even play, just see the behavior of enzymes. I am going to teach you today how they help each other. That is the feedback inhibition. Their cooperation reduced their energy. If they will not cooperate with each other, they have to do a lot of work. You will see that, for example, 11 players are there and 3 players are not 3 players are little ill, but other players are working very hard, so definitely team still will win. There are some examples when some players were shown red card by umpire in top football matches, and still the team with the 10 players won, because they had motivation, because they had coordination, they had teamwork, they were together. So that is the point I am going to discuss you today. Look here, these are the friends who are working together. Look at the team. So I am showing you the team of enzyme. You have seen the teams of players. You have seen the teams of many departments. Now I am telling you the team of enzymes. One member, enzyme I, coach of the team. Enzyme number 2. Look here. You are watching the figure, so I am just indicating one by one the each member. So third member is in front of you, enzyme number 3. This is enzyme number 4. So 4 enzymes are working to catalyze this chemical reaction. Look here, their friendship relationship. If they will, enzyme 1 will do the function of enzyme 2, and he will not be agree, no, I will bat, I will not bowl, I will bat because I want batting. This reaction will not be catalyzed. So enzyme 2, if he tried to do the work of enzyme 3, and they all will do at specific time, that is astonishable. That is astonishable. I was drinking water. Look here, enzyme 1, if do the function in 6th minute, enzyme 2 will do the function in 7 minutes. If enzyme 2 do the function in 5 minutes and enzyme 1 do the function in 6 minutes, the reaction will not occur. Timing. You are just standing near the goal, you are waiting for the ball came, you was not active, the ball gone, you are very active. Now the time gone. All players will be worried, all players will criticize you, the TV media will criticize you, the TV replay will tell you that you was not the man active during when ball came. These enzymes work when the ball came. These enzymes work when time is there to convert one reactant into the product. So their timing is important and their sequence is important. So enzyme 1 finished the work. The point I just wanted to convey. Enzyme 1 finished the work, enzyme 2 start the work. Look at these chemical molecules of protein. They are telling us many lessons. They are telling that you are alone, you are nothing. You are alone, you are nothing. And when you are in the form of the group, you are in the form of the team, you are the winner. If all Pakistanis are in one page, all Pakistan political parties are in one page, which international country will criticize Pakistan? Because we are the people that we are fighting in the Pakistan, so definitely we will get the negative image of the Pakistan to the other. So mean unity, as a unity, these enzymes working in the form of the team. And what is the advantage of working as a team? Look here, the reactant A, what is the name of the reactant? Substrate. What is the name of enzyme which can attach with the substrate? Active site. Inactive site of enzyme is called as allosteric site. The substrate attached with active site to form ES complex. I am just revising. What is the name of the structure of the protein? Globular structure. How many structure of the proteins are present? Primary, secondary, tertiary, quaternary. What is the quaternary structure? It is a more complex structure. The two type of the protein molecules present in the quaternary structure. Any two examples of quaternary structure? Hemoglobin enzymes. Why enzymes are more complex? Because they consist of quaternary structure. Enzymes work at specific pH. Enzymes work at specific temperature. The temperature at which the enzymes work maximum, that is called optimum temperature. The enzymes work at maximum pH. That pH is called optimum pH. So I was telling you some points. Proteins are composed of amino acid. The bond between amino acid peptide bond. Protein is polymer. Amino acid are monomers. The monomers combine to form the polymer. Water will be released. Name the process: condensational polymerization. Who forms the protein in the cytoplasm? Ribosome form the protein in the cytoplasm. How many units of ribosomes are there? 2 units, sir. Small ribosomal unit, large ribosomal unit. They are gathered or they are not gathered. They are separated or they are gathered. They are separated. They will not gather. They will gather only when messenger RNA will come. From where the messenger RNA will come? From the nucleus. From the nucleus. What is the boss? DNA is the boss. Who gives message to messenger RNA? DNA. Process name transcription. Messenger RNA come through which tube? Endoplasmic reticulum. What is the target of messenger RNA? Goes to ribosomes. Ribosome consists of which RNA? Ribosomal RNA. What the ribosomal RNA will do to form the protein? How it forms it? How it will form the? It will give the. How it will form the protein? It will give order to transfer RNA to bring specific amino acid ordered by DNA. So this is the arrangement. I must cofactors help the enzymes. Type of cofactors you forgot. You forgot the type of the cofactors. I repeat. Okay, I tell you. The one type is activator. They will not attach with active site and they are detachable. They will facilitate formation of ES complex, but they will not attach with active site. The prostatic group attach with the active site, but they will attach by covalent bond. The third one is the coenzyme. They will also attach with active site. We have to make very key attention, making difference between prostatic group and coenzyme, because technically they both are similar, because they both are attaching with the active site, but one is attaching with the covalent bond, one is attaching with the weak linkage. With the weak linkage, that molecule attached, that is called as covalent coenzyme. Weak linkage coenzyme. The covalent bond, the molecule who form the covalent bond, that is the prostatic group. Go back to this one. The initial substrate. This is the initial substrate. It goes to the initial substrate, means the main substrate reactant. It goes to B. First of all, I should tell you that what is the chemical reaction. E is the product. Okay, E is the product. Unfortunately, a molecule cannot go directly into E. A molecule cannot go directly into E. So what will happen? A can go to B, B can go to B, C can go to D, D can go to E. So these are series of chemical reactions, and each reaction enzyme is specific. These enzymes are working as a team and they are working in a timing. Their timing, one reaction complete, A to B reaction is catalyzed by enzyme number 1. This reaction is catalyzed by enzyme number 1, and then what will happen? The second reaction then enzyme to start working. These are the intermediate substrates, but they are working in a timing. Yes, in a allotted timing. Enzyme work, finish the work, B is formed, then start B to C formation B2C formation start. Similarly, this enzyme to work after enzyme 1. Look at the team, how they are working. The point I was talking to you, the enzyme, the intermediate molecule C. Now that will be catalyzed by, you are watching the figure, I am indicating the enzyme C will be converted into D, capital D. Which enzyme? Enzyme 3. It means that these are intermediate molecules, these are intermediate substances, and enzyme D changes to, and the substrate D changes to substrate E by enzyme number 4. These all enzymes work alternatively at their specific timing. Mean enzyme 3 finish the work, enzyme 4 starts the work. If enzyme 3 finish the work and enzyme 4 start converting D to C, D is not available, so from where he will convert the D to C? The same example I was telling you, when you are batsman and ball is coming, when ball will be closer to bat, you have to hit. When ball gone to wicket keeper, you are hitting, your eye hit the ball did not touch the bat, the ball was gone to wicket keeper, my son. You have to see the ball. So enzyme is watching the ball, enzyme is watching the substrate. When D is formed, convert to E. The feedback inhibition did not start, the process did not start yet. The process still did not start yet. I will tell you what is the process. This enzyme E, when final product will be formed, when the final product will be formed, mean enzyme E will be formed, final product will be formed, and final product will give message to, final product will go, and if I can do this one, then yes, this will be a good idea to show you. The final product for E will go to enzyme number 1. This is a strange meeting. This is a strange meeting. You are going to meet the villagers of other village, which is not concerned with you. Enzyme number 1 is not concerned with the product. Enzyme number 1, it is not concerned with this reactant and product. But this product E is going to enzyme number 1 and it attach with allosteric site, mean it says to enzyme, don't do this chemical reaction, don't convert A to B, because enzyme number 1 A will not convert A to B, the B may be not converted to C, C not converted to T, D not converted to E, the E will not be formed. Why enzyme E was gone to, why product E gone to enzyme number 1 to tell that I am formed, no need to form the E. You are getting? This is self-control by the product. Product is controlling, I am formed much in amount, like insulin, I am formed in much in amount and glucose level is decreased. Now don't form me, Mr. Enzyme number 1. Enzyme number 1 will say, I am not farming you, gentleman, I am farming B. You are E and you are coming from 4th village to me and telling that you are guiding me that I should not do. Why I will not be? I farm B, I am not farming you. E will say, no. When you will farm B, B will be changed into C, C will change into D. Your all friends will be in trouble. Enzyme 1 thing, oh, you are right, our all team will be in trouble. It means we will have little rest when I will not form B. E is formed. So that is the way by which enzymes will stop work when output is achieved, when the product is formed. This is called feedback inhibition. The topic I told you, feedback inhibition. Feedback inhibition. Feedback inhibition is the process in which products control the reactant itself. The product E went to enzyme number 1 to control. So we are coming with the new discussions that what are the factors who can increase rate of reaction. You did not know, because if you are expert of biochemistry of the human body, if you are athlete, if you are a worker, if you are a student, if you are a player, you will always think to increase rate of enzyme action. Lot of medicines are in the market which increase the rate of reaction. Lot of doctors are advising to increase the rate of reaction. What is the advantage when enzymes are when enzyme reactions will be more? Everything, every part of your body will be more active. So upcoming discussion how you can increase the rate of enzyme action.